DNA structure and replication. A genetic material must carry out two jobs: duplicate itself and control the development of the rest of the cell in a specific way. A brief history of DNA. Friedrich Miescher termed phosphorus and nitrogen contain acidic substance with a nuclei of white. Gort first to link human inheritance and protein inborn error of metabolism in 1902. Friedrich Graef demonstrated that the transforming principle could be transformed between different bacterial strains. A Diplococcus pneumoniae infection in mice. Mice develop pneumonia and die. Two types of pneumonia, rough and smooth. The smooth is virulent. Heat killed smooth, virulent strain. Avery and McCarthy showed that DNA, not protein, was the transforming principle in 1944. The experiment, rough, non-virulent strain, and plus the heat killed smooth virulent, plus protease and plus DNAs. Hershey and Chase used viral infection E. coli to show that protein doesn't transform genetic information in 1953. DNA is the genetic material. Viral protein code, radioactivity labeled. Virus infect bacteria. Phipposlevans identify 5-carbon ribose and similar desoxyribose and found that component of the nucleic acid sugar nitrogen base pair. What is DNA? DNA is desoxyribonucleic acid composed of phosphate, desoxyribose sugar, and different nitrogen bases. RNA ribonucleic acid. Here we find the base pairs, purine, ibermethylation. Edward Chergaff showed that DNA from several species contain equal amount of A, T, and equal amount of C and G. Base, sugar, phosphate. What evidence did they have at the time? Base and sugar and phosphate. Here, the Rosalind Franklin and its picture, X-ray picture of the DNA. Watson and Crick and their model for the helical structure of the DNA. Double helix. DNA consists of two chains in right-handed double helix. The diameter is 2 nanometers. The two chains are anti-parallel. Sugar and phosphate backbone connect nucleotide in a chain. DNA has directionality. Two nucleotide chains together bind into the helix. Hydrogen bond. Here the desoxyribose, the carbon atom on the sugar ring, 5 and 3 hydroxyl group, the model of the double helix. Watson and Crick explained one from DNA called B form. At least two other forms are known to exist. Other forms and the extension of DNA. The extension when stretched DNA of smallest human chromosome, 14-meter chromosome during replication. Here, how DNA composes itself inside the cell. DNA around the histone and other proteins and chromatin structure, nucleosome, 8 histone proteins, non-histone, and chromatin. Here we find the DNA and how it bound itself inside the cell. Is 2. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Watson and Crick. The semi-conservative model, the conservative model, and the dispersive model. The ultracentrifugation of the DNA. Double-strand DNA unwind. The junction of the unwind molecule is replication fork. New strand is formed. Helicase unwind the parental bind. Proteins primase add short primer. The steps of helicase bind to DNA sequence. Binding protein prevents single strand from rewind. polymerase add DNA nucleotide to RNA primer follows in 5 to 3 direction. DNA polymerase enzyme add DNA polymerase proofreads base added the replace incorrect nucleotides. Leading strand synthesis continue in 5 to 3 direction. Discontinuous synthesis produce 5 to 3 DNA called Okazaki fragments. Leading strand synthesis continue in 5 to 3 years. Endonuclease enzyme remove RNA ligase form a bond between sugar phosphate